talk about question number 9, and this question is asking briefly mention the contribution of T.H. Morgan in genetics. So basically in the study of genetics we study Mendelism. The first part of genetics is about Mendelism, that is a scientist Mendel, and the second part of genetics is about T.H. Morgan. This scientist T.H. Morgan has contributed in the genetics very much, and the topic linkage and recombination is from this scientist only. So first of all, if we talk about the contribution of T.H. Morgan, what he has contributed in the genetics, as I mentioned already, that he contributed or he proposed the concept of linkage and recombination. Basically the concept of linkage actually introduced by another scientist such as Sutton and Boveri, but the experimental proof was given by this scientist named as T.H. Morgan. Now as we know that Mendel performed many experiments in genetics on which plant? Pea plant, Pisum sativum. He chose the Pisum sativum for his experiments. Whereas if you talk about this scientist T.H. Morgan, he chose Drosophila, which is a fruit fly. Drosophila melanogaster is the complete scientific name for fruit fly. He chose Drosophila melanogaster, named such as fruit fly, for his experiments. Alright, so T.H. Morgan introduced linkage and recombination. T.H. Morgan described linkage as the presence of or coexistence of two or more genes in the same chromosome, which are lying on the same chromosome and lies close to each other, which are going to inherit together into the progeny. These genes are named as linked genes by T.H. Morgan. He also performed dihybrid crosses in Drosophila to show that linked genes are inherited together into the progeny and are present on X chromosome. By the dihybrid cross performed in the Drosophila melanogaster by the scientist T.H. Morgan showed us that linked genes are inherited from generation to generation together and are present on the sex chromosome, that is X chromosome. Alright. Also he described that the loosely linked genes show high percentage of recombination, whereas the tightly linked genes on the chromosome show very low percentage of recombination. So this is the contribution which is given by this scientist T.H. Morgan into the genetics. So now let's write the answer of question number 9. First of all, we will write T.H. Morgan worked on the fruit fly such as Drosophila melanogaster. Drosophila melanogaster is the scientific name for fruit fly, melanogaster. Drosophila melanogaster, to show his results, he chose Drosophila melanogaster, a fruit fly, for his experiments. He introduced the concept of, actually he does not introduced, he proved the concept of linkage. He proved the concept of linkage and recombination. Alright. He also performed he performed, how does he actually proved the result of linkage? He performed the dihybrid cross. He performed dihybrid, and because of this dihybrid cross done on the Drosophila melanogaster proved about the linked genes, about linkage. He performed the dihybrid cross on Drosophila, to show that the linked genes inherit together into the progeny or into the next generation, into the progeny or next generation, and are present. He also proved that the linked genes are present on the X chromosome. So this was the result given by the scientist T.H. Morgan. He also described first of all, he described the coexistence of two or more genes on the same chromosome, lying close to each other, are linked genes, and because they are linked genes, they will be inherit into the progeny together. And also he said that the tightly linked genes show low recombination, while the loosely linked genes show higher percentage of recombination. So this is the contribution of T.H. Morgan into the genetics, and this is the complete solution of question number 9.